Good evening, everyone. Welcome to the Snyder Institute for Chronic Diseases Mini Medical School Lecture Series. We are pleased to host a compelling presentation and discussion this evening on the topic of mitochondrial dysfunction in disease. My name is Hannah Riley, and I will be the program host for this evening. Before we get started, I would like to go over a few housekeeping items. There'll be two parts to tonight's program. First, each of our presenters will speak for approximately 20 minutes, and then we will open the floor to audience questions. We will take a 10-minute coffee break between each speaker, and refreshments will be provided to those of you here in person. You're welcome to ask your questions at any time during the presentations using Slido. To access Slido, you can either scan the QR code up on screen or type slido.com into your browser and manually enter the event code Snyder MMS into the participant field to post your question. Of course, if you prefer to ask your question in person, you'll have the opportunity to do so as well. Questions submitted to Slido will be viewable to all audience members, and participants can upvote the questions they would most like to see asked. Now on to more specific housekeeping items. For those of you who have joined us online, if you lose your internet connection and drop out, you can return to the session using the same Zoom link you used to join us at the start. If you experience any technical issues, such as you cannot hear or see the presenters, please email snyder@buxa.com and our team will do their best to support you. For those of you here in person, we ask that you now switch your cell phone to silent mode. Please note this event will be audio recorded and the recording will be posted on the Snyder Institute website. To begin our program, the Snyder Institute at the University of Calgary's Cumming School of Medicine, located in the heart of southern Alberta, would like to acknowledge and pay tribute to the traditional territories of the peoples of Treaty 7, which include the Blackfoot Confederacy, comprised of the Siksika, the Piikani, and the Kainai First Nations, the Tsuut'ina First Nation, and the Stoney Nakoda, including the Chiniki, Bearspaw, and Goodstoney First Nations. The city of Calgary is also home to Métis Nation of Alberta Region 3. Now I'd like to offer the floor to Dr. Derek McKay, who will provide a brief overview of the Snyder Institute.
Okay, so this you'll find on our website. Everything that happens within the Institute is very discovery based. I think we're renowned for discovery research, the addition of new knowledge, but really that's all in line with making discoveries that's going to improve the lives of individuals living with chronic or infectious disease. And these are major killers right now. You can think of chronic disease as modern plague. So all of our work is ultimately aimed at improving the lives of individuals who suffer from chronic and or infectious disease. Just a little bit of promotion for the team. Kamala here, you can see on your right, is our co-director. Skylar runs the business office. And then the team that makes everything click is the office personnel, Holly and Hannah, who you met earlier, Ronman, who was giving out the books at the door, Anurag and Kelly. And I'd like to thank them for all of their efforts and dedication that make this and many other community engagement activities possible. Yep. As I always like to say, and in terms of the discovery research, it's people that really are our number one resource. We work very hard at recruiting world leaders, early career investigators, and more senior individuals to really drive innovative discovery research. The team is fairly big at around 500 individuals. We have 125 or so faculty members, so assistant professors through to full professors, 60 postdoctoral fellows. The real workhorse within the group and deserve, if not all of the credit, are the trainees, the postdoctoral fellows, and the graduate students, and then some staff that fill our core facilities. And the goal really of the office is to create a research environment where individuals can excel. You'll see in the next slide, it's a real breadth of research and disease activities that we look at. So we're looking to create that environment that allows these very talented individuals to reach their potential and to really excel, and to create an environment of respect and diversity where everyone feels welcome, their opinions are listened to, their opinions are acted upon, and that really creates a very vibrant research environment. And we also feel very passionately about that we are socially accountable. It's one of the reasons we're very proud of Mini Med School, is it gets us into the community so that the Calgarians can understand why we're here and what we're trying to achieve. There are 6 major groups within the Snyder Institute. So we have a bacterial research group, one on immunology, an airways health research group, a GI or a gut research group, inflammation, which covers many other sort of target organs, and then a smaller group focused on kidney disease. So these would be like the 6 major pillars when we think about at least chronic diseases, as well as the infectious burden as well. This list is not complete, but you can see it's incredibly broad, with representing the expertise within the Snyder Institute, conditions, model systems, cell lines, all aimed at understanding and advancing knowledge of asthma, inflammatory bowel disease, fungal infection, parasitic infection. The list is almost endless as we try to make inroads into better treatments in many of these chronic problems. We introduced this research platform about 18 months ago. This is where we grow human organoids. So the basic idea here is we can take samples from a healthy individual or a person with some particular condition, and from their blood cells or from that tissue, we can grow mini organs representative of that diseased organ. So we can take pieces of colon here, you can see here, and grow these little mini guts in the lab. And that allows us to test drugs on these organoids, to understand how they grow, differentiate, how bacteria influence their development and their behavior. And would this then lead to increased awareness of disease processes and the development and the identification of new therapeutic targets? Great expertise in the gut. We're growing these little mini kidneys, and emerging expertise in lung organoids as well. Of course, you probably will recognize this if you're a regular attender. We're very proud of the International Microbiome Center. This really is the biggest facility of its kind anywhere in the world. A remarkable facility where we're trying to understand the complexity of the microbes that live on us and in us, how these contribute to normal function, and when something goes wrong, how this then can precipitate disease. Live cell imaging. This is really maybe the foundation of the Snyder Institute going back 12, 14 years ago. Really world-class experts in imaging immunity in real time, in tissues, in the gut, in the airways, the interaction of those immune cells with various pathogens. This is state of the art. The group that leads the live cell imaging suite does an annual course on high resolution imaging. And you may see some images from Dr. Shutt later. This course is always oversubscribed. People from across the country and North America are queuing up to get the educational benefit from the live cell imaging personnel. I'll close with 7 slides of some of our achievements. These actually, I think, were on the rolling slides, so you might recognize them. Perhaps those at home didn't see them. But Dr. McDonald and his team did some groundbreaking work about 8 months ago, looking at the interaction of the microbiome with a particular type of immune cell, the neutrophil, in patients in the ICU, and defining that those neutrophils were doing something unusual and they were not functional, and that was leading to more severe outcomes in those individuals. So again, allowing us to try to tease apart how the bacteria and the neutrophils, this type of immune cell, in a critically ill patient is affecting the outcome of that condition. Christoph and Manon have been repurposing drugs to look at better pain management strategies. Pain is a huge symptom in many chronic diseases, maybe the first one that most patients will complain about. So trying to make inroads there in symptoms that are of critical importance to the patient. Laura and Karen working with preterm infants, trying to figure out how the microbiome will affect the development of those preterm infants and their susceptibility to a variety of chronic diseases. We have Rachel in Dr. Kubz's lab, who did some really stunning work. This work took about half a dozen years, give or take, and was published earlier this year. A novel mechanism of how macrophages, again, this a special type of immune cell, can promote wound healing. Brian and Nicole, once again, working with their state-of-the-art imaging equipment, looking at fungal diseases in airways. So in the patients with the pneumonia and other airways conditions, are the fungi that get in there causing problems, and identify that, again, there could be a defective immune cell allowing these pathogens to get out of control. And then maybe more at the community level, John Connolly, some of you will have heard John speak at this session. John's a world-renowned clinician in infectious disease, and him and colleagues have put together an app that's available for physicians and veterinarians, really to look at antibiotic resistance, which is a huge problem and will continue to be a problem if we don't come up with additional drugs or therapeutic modalities. And then finally, Mike Parkins and his colleagues, very interested in water quality, water control, putting in place a citywide survey or surveillance system to look at drugs in the water, medications in the water, and the outbreak and spread of infectious disease. So a very practical application of the research being done in Mike's lab. So with that, once again, let me welcome you. Thank you for being here. I hope you enjoy this evening. Thank you so much.
Thanks, Derek. I'd now like to introduce this evening's moderator, Arman Mohan. Arman is a PhD student here at the University of Calgary that's supervised by tonight's speakers, Dr. Tim Shutt and Dr. Derek McKay. His project examines how mitochondrial damage dysregulates inflammatory signaling in the context of inflammatory bowel disease. Arman is also actively involved in the community, acting as the programs and education co-lead of the Calgary chapter of the Crohn's and Colitis Canada, and is the vice president of his departmental graduate association. Now I will hand things over. Take it away, Arman.
Good evening, everyone, and thank you for joining us. So I want to thank each of you for your continued support of our mini medical lecture series. I'd also like to thank our sponsors, UCalgary's Cummings School of Medicine and AstraZeneca, for their continued support of our mini medical school lecture series. Finally, I want to thank Snyder Institute members and collaborating institutes for volunteering their time to present such excellent lectures. Without their support, there would be no mini medical school series. And now I am pleased to introduce our first speaker of the evening, Dr. Tim Shutt, PhD, who is an assistant professor in the departments of biochemistry and molecular biology and medical genetics, and a full-time member of UCalgary's Snyder Institute for Chronic Diseases, Hotchkiss Brain Institute, and Alberta Children's Hospital Research Institute. Dr. Shutt's work focuses on investigating interplay between mitochondrial dynamics, cellular stress, and the regulation of mitochondrial DNA and the genetic causes of mitochondrial disease. Welcome, Dr. Shutt.
Right, thanks, Arman, and thank you everyone for showing up tonight and hopefully getting the opportunity to learn a little bit about mitochondria and their role in disease. Before I get into the disease side of things, I just want to start with some basic background and overview of mitochondrial function, because it's likely that not everybody knows quite as much as I do when I'm studying, thinking about mitochondria every day. So the first thing I want to ask the audience is, what's one thing you know about mitochondria? Anyone? Powerhouse of the cell? I think that's what I heard. And you're absolutely right. That's one thing just about everyone knows about mitochondria, if you know anything, is that mitochondria are the powerhouse of the cell. So in that regards, the key role that mitochondria play in making energy in the form of a molecule called ATP. And they do this through a process called oxidative phosphorylation, which is sort of outlined down here. I'm not going to go into the molecular details because it's fairly complex and I don't want you guys to fall asleep tonight. Instead, just to highlight that, yes, mitochondria are important for energy. That's without a doubt, a fact. But hopefully I'll impart upon you a few other important things that mitochondria do. And hopefully you'll, at the end of the talk today, know a little bit more about mitochondria and why they're important for health. So another aspect of mitochondria, most people, if you see a textbook picture of a mitochondria, it's this sort of bean shape that you often see inside of the cell. In actual fact, if you look at a cell, the mitochondria form a network, which you can see here in purple, which is distributed throughout the entire cell. So it looks nothing at all like these bean-shaped structures that you might see in a textbook. Beyond that, mitochondria also encode their own genome or mitochondrial DNA. And so what you can see here, all these yellow spots are a copy of the mitochondrial DNA. And so you can see within the cell, you've got hundreds of copies of this mitochondrial DNA that are distributed throughout this mitochondrial network. And so one of the reasons I'm talking about the network is in part because it's something that my lab studies and I'm really interested in, but it's also really important for how mitochondria function. And so the shape or the morphology of this network can change in different cells. Some sort of basically a spaghetti type morphology where it's all big long strings all attached together, down to just individual puncta, which are maybe a bit more what you might think about when you think about those beam sort of shapes that you see in a textbook. And my lab is interested in studying the processes that change the shape of these mitochondria. And the shape is really important for determining the function and how well those mitochondria make energy, as well as some of the other things that mitochondria do. To give you a bit more of a aspect of what this looks like in an actual living cell, I'm going to show you a video here of mitochondria. And you can see that those mitochondria are going to be moving around in the cell. And you'll watch here, you'll see this one mitochondria is going to come over, visit his friend, and they're going to fuse together. And so this is happening all the time in our living cells. They're constantly moving around, dividing and fusing. And so I find that really fascinating, which is one of the reasons I study it. But it's also an important process. When this doesn't work properly, your mitochondria are dysfunctional. And so beyond the fact that mitochondria have a role in energy, what did I do? There we go. I just want to highlight another take-home message is that mitochondria are much more than just the powerhouse of the cell. So this slide is just trying to illustrate many of the different roles that mitochondria have. So obviously generating ATP, or the energy that our cells use, is key and critical. Beyond that, mitochondria are involved in a whole host of different metabolic processes in the cell. And this includes things like making nucleotides, which are required for DNA. And we need those nucleotides if the cell is going to replicate. In addition, some of the signaling molecules, for example, steroid hormones, those are all regulated and controlled by mitochondrial function. Another key aspect here is apoptosis. That's cell death, or a specific type of regulated cell death. So not only do mitochondria control when cells are dying, but they're also, as I mentioned before, involved in making these key components for replication. So mitochondria are controlling both the replication or the birth of new cells and the turnover or death of cells. Another example that people might be familiar with is reactive oxygen species. And so mitochondria are a key spot for the generation of reactive oxygen species. Most people have heard about these in the context of damaging molecules, but these reactive oxygen species can also be important signaling molecules in the cell. All right. Okay. So why are mitochondria so important, and why are they involved in so many different processes in the cell? And so for that, we're going to turn back the page about 1.5 billion years and look at the origin of mitochondria and where they came from. And so we know that mitochondria actually used to be bacteria. And these bacteria were taken up into cells, and they, through what we call an endosymbiosis process, have evolved into the mitochondria that we recognize today. And so these bacteria, or which we now recognize as mitochondria, have been involved in this symbiotic relationship with the cell for over 1 billion years. And that's in part why they're involved in all these different processes and decisions that the cell makes. And so the other thing to sort of, I'll emphasize here in the context of mitochondria being really just fancy bacteria, is the fact that I like to think of mitochondria as being the original microbiome. And I'm sure if you come to the rest of these talks, you'll hear more about the microbiome later on and a number of other ones, but just remember mitochondria were the first. So the bottom line here is that mitochondria are really complex. They have many important functions in addition to just generating energy. They're very intricately regulated. And what this means is that there's lots of different ways that mitochondrial function can go awry. And at the end of the day, what we're going to sort of talk about next is that this dysfunction of mitochondria can lead to disease, right? So hopefully now you get a bit of better picture of what mitochondria do and why they're important. And now we'll talk about what happens when they don't function properly. So mitochondrial disease is a rare disease. It's considered, a rare disease because it's estimated about 1 in 3,000 people will have a mitochondrial disease. Mitochondrial diseases, just like mitochondria, are complex. They're considered multisystemic, which means that they can affect any tissue. They can develop at any age, although typically the more severe forms tend to present at younger ages in children, often before the age of 10. And so highlighted over here, you can see it. Basically, mitochondrial dysfunction can affect just about any tissue in the cell, but it doesn't always affect all tissues. And so that's part of the reason it's really complex. As you might expect, often muscle tissues and brain, which require lots of energy, can be severely affected by mitochondrial dysfunction. But on top of that, the mitochondria play basically an important role in every cell in the body. And so perhaps that's why it's not surprising that just about every tissue can be impacted by mitochondrial dysfunction. So mitochondrial diseases can be caused by mutations either in the mitochondrial DNA, so all those little spots that I showed you before, as well as mutations in the nuclear DNA. So mitochondria is these sort of little organelles in the cell, and mitochondria are made up of about 1,500 proteins. So that's about 1,500 genes which encode these proteins, and a mutation potentially in any one of those could cause mitochondrial dysfunction. The mitochondrial genome itself encodes only 13 proteins. So less than 1% of the proteins in mitochondria are actually encoded by mitochondria. The rest are all encoded in the nucleus and they get imported into mitochondria. And so these 13 proteins encoded by this mitochondrial DNA are actually all key core subunits of the oxidative phosphorylation system, which is required for energy. And again, I'm not going to go into much detail on that, other than to say that the DNA encodes key proteins here. And so the nucleus would encode the rest of these proteins. So classically, or I guess what we consider classic mitochondrial disease, often is associated with impaired oxidative phosphorylation or the ability to make energy in these cells. However, if mitochondrial disease was just about not enough energy, you'd expect that it would look the same in all patients, and that's certainly not the case. And so part of the reason we think that mitochondrial disease is so variable is that mitochondria do a lot of different things and different tissues are impacted differently, not just in the context of energy production, but in a lot of the other functions that I mentioned before as well. So honing in a little bit more on the mitochondrial DNA, because that's sort of one of the unique aspects of disease in mitochondrial disease. We know there's over 400 different mutations in the mitochondrial DNA that have been identified to date. And another interesting aspect about your mitochondrial DNA is it's inherited maternally. So you get your mitochondrial DNA only from your mothers, right? Our nuclear DNA, you get one copy of each gene from your mother and one copy from your father, right? So you have basically half and half. Mitochondrial DNA, on the other hand, is exclusively inherited from your mitochondria or from your mother. And so that's going to have important implications also in the context of inheritance of mutations on the mitochondrial DNA. And so another aspect that's different with the mitochondria is the fact that you have hundreds of copies in each cell. So if you have one copy of your mitochondrial DNA that gets a mutation, does it really matter if 99% of the rest is still healthy and good? And so this leads to a situation that we refer to as heteroplasmy, which is basically a mixture of wild type and mutant mitochondrial DNA within each cell. And so that's sort of shown down here, as you can have different levels of mutant mitochondrial DNA here in red, and the proportion of those mutant mitochondrial DNA within each cell will influence the mitochondrial function, and that will influence not only the severity of the disease, but even the presentation or the type of mitochondrial disease. And so typically, once you get levels of mutations in the cell up above 50% and higher, you start to exhibit sort of more severe mitochondrial dysfunctions that can lead to disease. This is also important in the context of the inheritance of the mitochondrial DNA, because you can have a mother who has a low copy of a mitochondrial DNA mutation and doesn't really have any disease at all, but she can pass on higher copies or lower copies, and that's distribution during generations is basically sort of random. And so you don't always know when somebody's going to be getting a mitochondrial disease because of the mitochondrial DNA, because the mother might be perfectly healthy and perfectly fine. And so that also makes it a little bit more difficult when you're trying to figure out the genetics behind a patient's disease and what's going on. So beyond just changes in the mitochondrial oxidative phosphorylation and energy production, we now know that mutations in a lot of the other important functions in mitochondria can also lead to disease. So for example, the proteins in the mitochondrial DNA need to be transcribed and translated. If those processes are messed up, it can lead to disease. Likewise, the replication of this mitochondrial DNA, there's a reason we have so many copies. If you don't have enough of them, that can also be detrimental. The fission and fusion events that determine that shape and morphology of the mitochondria, if those get impaired, that can lead to disease. Reactive oxygen species, too much reactive oxygen species can be damaging and also be detrimental. That also, I guess, another one aspect here is repair of the mitochondrial DNA. The repair capabilities of mitochondrial DNA are limited compared to the nuclear DNA. So in addition to being close to reactive oxygen species where they're made, they're also more sensitive because they can't fix their DNA quite as well as the nuclear DNA. And then finally, the last aspect here is called mitochondrial autophagy. Autophagy is a way that the cells, I guess, basically recycle their damaged or used components. Mitophagy is a specific type of autophagy where it's important in regulating the turnover of damaged or older mitochondria. And so one clear example here is we know a lot of the key players that regulate this mitophagy or clearance of damaged mitochondria are implicated in Parkinson's disease and other neurodegenerative diseases. So in the context of diagnosing mitochondrial disease, this can be extremely challenging, in part because it's hard to define what mitochondrial disease is in the first place, in part because it's so complex and because presentation can be so variable from patient to patient. Even patients that have the same mutation can have different presentations sometimes. And then on top of this, it's rare. So most clinicians won't see this on a day-to-day basis, so they're not necessarily going to know what to look for or be able to recognize it. In this context, one of the issues of mitochondrial disease diagnosis is the so-called diagnostic odyssey, where patients can have something that's wrong with them and they go to the doctor for 2 or 3 years before they figure out what might be the problem. So in the last 10, probably maybe 15 years, there's been a huge change in the diagnosis of rare diseases, such as mitochondrial disease, where they've started to take advantage of genetic diagnosis and the ability to sequence basically your entire genome. And so this has really sort of allowed, at least at the bioinformatics side, trying to identify what is the specific mutation in a person which is causing their specific disease. So this is what we call as a genetic diagnosis. And so what happens when they do this next generation sequencing and diagnosis is there's sort of 3 outcomes. You can get a clinical diagnosis where you find a known disease variant and the patient phenotype matches with what's been described before. So this would be considered a solved case based on a genetic analysis. Unfortunately, this only happens about 1/3, maybe half the time. The rest of the time, what happens is you'll get either no obvious candidates that come out of this analysis, or you get these variants of unknown significance. And these are variants that might be causing the problem, but nobody's seen them before, or they're in a gene that hasn't really been characterized very well. And so it's not really clear whether these candidates are what's causing the disease or not. And so one aspect of the research in my lab is working with clinicians when they find these variants of unknown significance in their patients that they think have mitochondrial disease, or if they get a variant in a gene which is involved in mitochondrial function, they'll come to me and we'll work together with them and try and figure out whether a specific variant might be leading to the disease in that specific patient. And so this obviously is not an easy task and it takes quite a while. the first part here, with the next generation sequencing, we can actually go now within about a day of taking a patient's DNA, sequencing it, and getting candidate variants. And so that's sort of the top end of how quickly things have gone now. Used to take several months, but now in extreme cases, they can do it in as little as a day. So it's really amazing how far that's come lately. So at the end of the day, diagnosing mitochondrial disease and having this genetic diagnosis is really important. It identifies a causative gene, and this can provide answers to families who've been searching for sometimes years as to what might be wrong with a particular family member or patient. It also can inform family planning decisions as far as the genetics and whether a family might decide whether they want to have a family in that context. It also can inform the doctors and clinicians as to what treatments might be or what the disease progression is going to look like over time. With respect to treatments and mitochondrial disease, we really don't have any good treatments for mitochondrial disease yet. Most of the options are just trying to manage things, and there's no cures at this point for mitochondrial disease. In that context, when we end this diagnostic journey for the patients, on the lab side of things, we're really just sort of beginning a mechanistic journey where we're taking these variants and taking the genes that they affect and trying to figure out mechanistically what's happening in the cell, what's happening in the mitochondria, what type of mitochondrial dysfunction is happening there. And at the end of the day, we need to really understand what the problem is in order to eventually be able to sort of try and develop a solution or potentially a cure or treatment down the road. So beyond classic mitochondrial disease, which tend to be more severe in this context and have a much more clear, obvious mitochondrial dysfunction, we also know that mitochondria and mitochondrial dysfunction is implicated in a growing number of diseases and disorders. And so this sort of diagram here is just sort of showing in the middle on these darker green colors are so-called classic mitochondrial diseases. So these are more specific types of mitochondrial disease that I didn't talk about in general, but the bigger circle on the outside, which might be difficult to see, and you don't need to worry about all the specifics, just to highlight that there's a growing list of diseases, cancer, cardiovascular disease, neurodegenerative diseases, inflammatory diseases, where we know that mitochondrial dysfunction plays a key role. And so by studying what's going wrong in these rare mitochondrial diseases, we're hoping that we can also apply that knowledge to these more common diseases, which also have mitochondrial dysfunction. And before I go, I just want to quickly introduce the role of mitochondria in inflammation, which is something that Dr. McKay is going to pick up in the second part of the talk here. And so if you remember, I told you early on that mitochondria are basically just fancy bacteria, right? And so what happens normally when bacteria or viruses get in our cells, it causes inflammation. And so the same thing is true when mitochondria are damaged. They can release components that basically look like bacteria, and those can induce inflammation in the cell, which can turn on inflammation in the entire sort of tissue or organism that's being affected. And so that's just sort of the first example of how mitochondria can be involved in promoting inflammation. Beyond that, they also have key critical roles in a lot of the inflammatory pathways in sensing viruses and other bacteria that get in the cells. So I don't have time to go into that in any more detail. But needless to say, mitochondria play a key role in inflammation, not only as being able to stimulate inflammation, but also in regulating the inflammation pathways once they get started in the cell. And so with that, we'll take a little break, I think, and we'll have coffee, and then we'll turn it over to Dr. McKay.
All right, welcome back. I hope you all had a chance to grab a snack and a drink. I would now like to introduce our next speaker, Dr. Derek McKay, PhD, who is the director of the Snyder Institute for Chronic Diseases, a professor with the Department of Physiology and Pharmacology within UCalgary's Cummings School of Medicine, and a former president of the Canadian Association of Gastroenterology. Dr. McKay is highly regarded for his research on unraveling communication pathways in the gut that control normal physiology and disease, both pro-inflammatory and anti-inflammatory mechanisms. He is a visionary leader who plans to maintain and build upon the established reputation of the Snyder Institute while fostering innovative chronic disease research, connecting with the university community, and leading education endeavors. Please join me in welcoming Dr. McKay.
Thank you, Armand. It's a pleasure to be back. It's been a while, so I hope you still recognize me. So in the next 20 minutes, I'm going to talk a little bit about my passion and how changes in the mitochondria can be linked to inflammatory bowel disease. And a lot of this really is driven from collaboration with Tim. I've found the way to be successful is to find someone much smarter than you and exploit them. So Tim, thank you for all of your expertise and helping my research program. And wrong way. There we go. In that vein, I think this is maybe one of the most important slides. All of the work, all of the research that gets done in the lab is by the trainees. I think all faculty members will tell you that they are incredibly indebted to their trainees, their passion, their dedication, their long hours in the lab. Many things not working, having to repeat stuff. So I always like to take the opportunity to thank them right at the start. And it's a fairly eclectic bunch from all over the world. Calgary's very well known for inflammation, disease-related immunities research. So a little bit of a mecca for trainees to come here. So in the next 20 minutes, here are sort of the messages of what I'm going to try to get through to make the case to you tonight that certainly mitochondrial dysfunction is in some way linked to inflammatory bowel disease. Is it an absolute cause? Maybe. Would it make things worse? I think almost certainly. We'll describe a little bit the impact of metabolic stress and how that affects epithelial barrier function. So you'll have seen from before, the epithelium that lines the intestine is a barrier between the outside world, what's in the gut lumen, the stuff that you eat isn't inside until it crosses that epithelial layer and get absorbed. And when that layer is leaky, that epithelial barrier is leaky, that can certainly precipitate disease. And metabolic stress or mitochondrial dysfunction will contribute to that. Not dwell too much on the processes of mitochondrial dynamics, these processes of fission and fusion. That's what Tim was talking about, the elongation, the fragmentation, very complicated molecular mechanisms behind those, but we'll talk about the implication of dysregulation. And then think about mitochondrial dynamics in the context of infection, one particular pathogen that might be relevant to inflammatory bowel disease, particularly Crohn's disease. Targeting mitochondrial fission, so that fragmentation process, can we target that to alleviate the signs, the symptoms, the severity of colitis, and then a little bit of a summary at the end to recap. So IBD, if you're not familiar with IBD, two major forms, Crohn's disease, ulcerative colitis. Right now, we do not know what causes inflammatory bowel disease. In the little cartoon to the right here, we put it at the nexus of a genetic predisposition, an uncontrolled immune response, some environmental triggers, but we really don't have definitive causes of why IBD develops, either Crohn's disease or ulcerative colitis. No known cause, no known cure. Probably right now, around 300,000 Canadians are suffering from inflammatory bowel disease. And this doesn't describe in any way the impact that those chronic conditions have on their family and their caregivers. These are very debilitating for the individual and very destabilizing for the family as well. Lifelong conditions, some IBD will arise very early in childhood, and with no cure, lifelong disease, multiple medications. Alberta is a hotspot for IBD. Canada is a hotspot for IBD. It's very much a sort of a westernized condition, but it is spreading worldwide. It's increasing in Canada. It's increasing across the globe. Our best drugs right now are biologicals. You may have heard of these. These are antibodies that neutralize aspects of the pathology. So the most popular one would be an antibody against TNF alpha, tumor necrosis factor alpha. That's our gold standard. That's the best therapy is available. That will work in maybe 50% of patients. So for 50% of patients, no useful therapy. Of those that respond to these biologics, the anti-TNF, 50% of those will lose that effectiveness. So that drug will stop working in that patient over time. So we really need better drugs. We need better treatments. So the researchers have to step up their game. We have to do better. And here's why. I think this really brings home the message. If you don't remember all the mitochondrial stuff, hopefully you'll remember the impact of IBD. So this is a case report from a colleague, Gil Kaplan, a 20-year-old woman, chronic Crohn's disease. In the past 5 years, every therapy that he's tried with her has failed. So they start on some anti-inflammatory drugs, steroids, moving up to the biologicals. Eventually, nothing is working. So there is no medication that's helping this young woman. Multiple hospitalizations, multiple surgeries, you can end up with short bowel syndrome. Courses of steroids. Steroids are really effective anti-inflammatory drugs. They bring horrible side effects, mood disorders, weight gain. They're not something you want to be on long term, and I'm sure the audience knows this. And during the time that this young woman was visiting Gil's clinic, she never really developed any durable remission, always had active disease. Abdominal pain, bloody diarrhea, chronic watery diarrhea, lots and lots of problems, not being able to leave the house. And ultimately, the solution at that stage was a colectomy. So to remove all of her colon, and now she lives with an ileostomy bag. And this young woman is 20. Now that sounds just horrendous to me. And then you start thinking, what are the impacts of this? So she was 20, so it started when she was 15. School interruptions. How's this young woman going to move into an early career? Great aspirations, always being affected by these chronic conditions. She can't deal with some of these. With most chronic conditions, there's psychological comorbidities, anxiety, depression. This is going to affect her. This is going to affect her family. Okay. And of course, not that I care so much about this, there's a huge healthcare utilization. I think the issue here is, how do we help the individual? How do we help patients suffering from this? The healthcare costs are an added bonus. And the very sad thing is, there's nothing really unique about this. The gastroenterologists here at the Foothills would pull dozens of cases like this. So this is really why we have to do better. So this little cartoon I showed you, this is the current state of the art, what we think causes IBD. So we started to think somewhat differently, influenced by Tim and his colleagues. If that barrier is really important to keeping stimuli on the outside that would stop the induction or driving inflammation, barrier function, it's an active process, it's going to be energy dependent. Would a lack of energy, would be perturbed energy, would mitochondrial dysfunction be allowing IBD to start or to push the progression of that condition? So really, the thought process is, if there's something wrong with the mitochondria, there'll be reduced energy. That's that ATP that Tim described. If you can't regulate the barrier, you're going to allow noxious substances, bacteria, bacterial products, weird food antigens, they get across that barrier layer into the tissue, activate immune cells, drive chronic inflammation. So we thought this makes sense. And there are many, many, many stimuli that will screw up mitochondrial function. Bacterial toxins, infection, some medications do it as well, right? Psychological stress, even smoking. So lots and lots of relevant, let's call them environmental stimuli that can change mitochondria. And when we look in the scientific literature, every single one of these, is it 8 factors, 9 factors, have been implicated in some patients with IBD as either triggering relapses in the disease, promoting the progression. This doesn't say it's via mitochondria. It says that things that can affect mitochondria have been implicated in IBD. So more correlation, not cause and effect, but giving us the motivation to try to test this. And when you look at tissues from patients with Crohn's disease or ulcerative colitis, evidence of mitochondrial dysfunction. Reduced ATP, so they're not capable of making normal amounts of that energy molecule. Swollen, irregular mitochondria, and you'll see this in the next slide. Mutations in genes that affect mitochondrial activity. And now, the gastroenterologists, the GI people, we have caught up with neuromuscular disorders and have started to take this serious and say, well, maybe there is a role for mitochondria in inflammatory bowel disease. And you'll find more and more review articles coming out trying to make that case. Is this something we've overlooked? And should we try to see how we can maybe develop new therapies for at least cohorts of individuals that have evidence of mitochondrial dysfunction? So we did some research, goes back a little bit, a very simplified model here. You can see we take this is a layer of epithelial cells. On this side, this would represent your gut lumen, so the outside of the body, and here's the inside of the body. And then we add in bacteria, stimuli, chemicals, drugs, anything we want. And we ask questions about the barrier properties of this layer. So what is coming across this layer due to these stimuli? A very simplified model system. So when we look at the passage of bacteria, if you put commensal bacteria, those good guys that hang out in the gut all of the time, if you put them on this side, then very few will go through, right? Because the barrier is tight, the commensals don't change that. If you take those commensal bacteria, put them on here, and now you add a drug that specifically messes up the mitochondria, then what happens is bacteria flux through here. So we've gone from 1 out of 3 to 18 out of 18, showing that when you mess up the mitochondria, the bacteria that should be happy living out here get access into this lower compartment. This would be the body. If you look at bacteria that actually get into the cell, so these are not bacteria that are invading, these are actively being taken up by the epithelium. And they shouldn't do that. That's not a good thing to do. They want to keep them on the outside. You mess up mitochondria, now you've gone over here on this side. Bacteria only, very few bacteria will get into the epithelial cell. If the mitochondria are damaged, that's a different signal. That's a danger signal. They start to take things up. And now many, many more cells will have bacteria inside. And the potential consequences of this, as these are danger signals. If you allow bacteria to cross the layer and get into the body, you can drive an inflammatory process. If you take bacteria that are meant to live outside the cell and let them get inside the cell, they're in the wrong place. And now they've gone from being a commensal to a threat. And that cell has to respond, and that cell will likely drive an inflammatory response because it's been invaded by a bacteria because its mitochondria are messed up. So now we're creating danger signals that are starting with mitochondrial dysfunction. That last slide was a cell line, something sort of artificial that we keep in the lab, but it works exactly the same way in tissue. So here, what we've taken are biopsies from healthy individuals. These are individuals undergoing colonoscopy for colon cancer screening. And we can mount these in the chamber and look at their barrier function. So this is the passage of bacteria across tissue isolated from a healthy individual. If you expose that tissue just to these commensal bacteria, every color and every shape is an individual person. And you can see, yeah, some will go across that layer. That's the normal response. But if you take parallel tissues from the same individual, so you can see the individuals are lined up here, damage the mitochondria. Could this be smoking? Could this be NSAIDs? Could this be psychological stress? If those mitochondria are damaged, now the commensal bacteria that don't really want to cross, don't really want to invade, they end up crossing that tissue, getting into the body. So again, cell lines, real tissue from healthy controls. So mitochondrial damage and dysfunction certainly can be a feature of IBD. Maybe not in everyone, but substantial numbers of individuals will have this. And we can induce changes in the mitochondria in our model systems, and that leads to barrier dysfunction, that disruption, things get leaky, allowing noxious things to cross. So the real question becomes is there a role for mitochondrial dynamics, that's funky movement of mitochondria joining and fusing in the barrier function? And can we experimentally target this as a proof of concept for drug development? So this is a little kidney bean that Tim talked about to make ATP. That's not real. They really are these beautiful networks. And here's some tissue sections from an individual, a healthy individual, an individual who's got mildly inflamed colon, and one that's severely inflamed. The blue is the nucleus of the epithelial cells that line the colon. The green is staying for mitochondria. Here in healthy, it's hard to make it out probably from the back. This is a very dense network interconnected throughout the cell, a little bit less towards the lumen, very densely packed mitochondrial networks. A little bit of inflammation. Now you start to see some of the networking because we're losing structure here. These things are falling apart. The apical side, very vesiculated, bloated, swollen, not so healthy looking, severely inflamed. We've almost lost all of the mitochondria here. So very substantive changes linked to inflammation. Is it a cause or effect? That's the big question. And I have to show you this because, where is it? Where's my movie? Tim showed you a movie. The only thing you have to remember is whose movie is nicer. These are cells in a dish that have been stained for the mitochondria. And once again, this really impressive remodeling. It'll finish in a second or two. This is obviously time lapse. So these are cells sitting in a dish, no stimuli, normal activity. How long do you think that movie was? So, did we capture for 1 minute, 10 hours, 2 days? Anybody want to take a guess? This very rapid dynamics, changing all the time. How long do you think that represents? Seconds? Well, this is actually a 13-minute movie. And it doesn't matter where things are moving constantly, right? And I've looked at this movie thousands of times. I just quite love this movie. So this was a paper that came out a few years ago, and the workers got really excited because they had found a new bacterium. It's an E. coli. They described it as a pathobiont. The E. coli by itself was a commensal, but something about the environment in a patient with Crohn's disease was turning that bacteria rogue, and it was now taking on the ability to damage cells. And they were very excited, and they published this, and all the red arrows moved a bit. These black dots inside this cell are the bacteria. So they've gone from outside to inside. They're now a pathogen. So the workers are really excited about this. And what they never mentioned was this, because they had no interest in this. These are really damaged mitochondria. So this got me excited, saying, well, were the mitochondria damaged that allows these bugs to come in, or did these bugs come in and damage the mitochondria? So we started to model that in our cell line, taking this prototypic pathobiont, an E. coli that has gone rogue, and say, what's it going to do to mitochondrial dynamics, that ability to fuse, rearrange, do normal activity. So this is an electron microscope picture, control cells, nice elongate mitochondria, and very healthy looking mitochondria. One's exposed to E. coli, swollen, irregular, they've lost their structure, very unhappy looking mitochondria, not a healthy looking cell. So this bug has disrupted mitochondria. One consequence, the tissue makes less energy. You can see here, when you expose to the E. coli, less energy. Here's another movie. You saw the last one. So these cells have been exposed to that E. coli, that bug gone rogue. And you can see, even to start with, no fibers, no long strands, just small, punctate, damaged, fragmented mitochondria. What does that mean to the cell? This is what we're trying to figure out. So if you stop it, this is the control. You can see these beautiful networks of the mitochondria in a control cell. If you add a commensal bacteria, he's one of the good guys, doesn't want to do any damage. We still have these lovely patterns and fibers, and those exposed to that pathobiont, that low-grade pathogen, has really knocked the stuffing out of the mitochondrial network. If you quantify that here in control, most of the cells you look at down the microscope under control conditions have beautifully fused mitochondria. Almost, half of the cells exposed to the pathobiont, these terribly fragmented mitochondria. Which is all well and good. Is this representative of the wider field? And we're not unique in this. Most of these are actually pathogens doing it. So pathogens do the same thing if you infect a variety of cells. Untreated cells, here's these lovely networks of mitochondria, and then you infect with the pathogen, it gets very vesiculated, broken up, damaged. We're quite excited because we're taking this very low-grade pathogen that doesn't do very much, and we're seeing that damage. And all's well and good. It's great to see that damage. Can we reverse it? Can we block it? Can we stop it? So these are human organoids. I talked about the organoid facility. So these are now real tissues from the colon of a healthy individual. This is work that Armand just did very recently and was published yesterday. Yesterday. Yeah. So here's our cell. This is the nucleus. These beautiful mitochondrial networks. Control. Expose them to our pathobiont, the E. coli. Once again, little fragments, vesicles, all busted up, not very healthy. And now he's treated them with this AR drug here. And when you come over and you do the morphometrics, you do the analysis, you can see with the E. coli alone, we're getting mostly fragmented cells. They look like this. When we add this drug at the same time, we're preventing that fragmentation and having more of an intermediate phenotype. So now we've got a way to say, if we know the pathogen can cause mitochondrial dysfunction in terms of disruption of the network, we can block that. And what does this AR drug do? So this AR drug, it's a synthetic drug, and it binds to a receptor on the cells. The natural ligand for that receptor is butyrate. So butyrate is made by the good bacteria. So it will bind to the cells and is helping maintain mitochondrial form and function. When individuals have their microbiome examined, if they have IBD, often the class of bacteria that disappear from patients with IBD are those bacteria that produce the short-chain fatty acids and produce the butyrate. So I would speculate that somewhere in their disease, when they lose those bacteria and the ability to make butyrate, they've lost that beneficial molecule that's helping preserve their mitochondria, which allows them to be hypersensitive to these pathobionts that'll destroy the mitochondria, they'll lose energy, the cell will die, the barrier will get disrupted, and we've started this downward spiral of creating disease. So would butyrate or this drug or variations of this drug be useful in the treatment and management of inflammatory bowel disease? In the last couple of slides, I just want to talk about a model of colitis. That was in vitro. That was with a sort of infectious colitis in a cell model. Can we use a model of colitis and again, target mitochondrial dynamics as a therapy? So the process is ridiculously complicated, but this drug is available not for prescription, but for research purposes. And this P110 drug prevents fission. It blocks one of the pathways by which mitochondria fragment. So when you have all those little punctate mitochondria, those long tracts and fibers of mitochondria are being cut up, and this drug has the capacity to stop that at least partially. So we did this experiment a couple of years ago. This is a murine model. So we're going to treat with our drug, give this chemical which causes colitis. So we can try to understand, can we block that? Daily drug administration, and then we're going to do an assessment of the severity of the disease. Here I've called this an illness score. So how healthy the animal looks. Do they have diarrhea? Have they lost weight? Are they moving around? Are they feeling ill? So our control animals, very healthy. The ones that got colitis, not surprisingly, they don't look so well. The ones that got this drug are doing substantially better. If we actually say, well, that's a macroscopic score, can we actually look at gut damage? What's the histopathology? Can we have a look at that tissue? The pattern, very, very similar. This is from controls, everybody's healthy. Here's our damage score. Over here, colitis, the tissue has evidence of ulceration, hyperemia, bleeding, damage. Substantially less damage if the mice had this 3-injection regime of P110, which will stop those mitochondria from fusing. So we don't know what cells are being targeted, but systemic delivery of this drug in this model system is limiting the disease severity. And what's maybe most important to the patients, as I talked about in the introduction, we can look at how the animals are experiencing pain. Are they uncomfortable? So the scale here is, this is worse pain, and here is just the pressure that's required to induce this in the mouse. So the DNBS, these are the animals with colitis, and you can see as you increase the pressure to cause pain in the abdomen, it gets it rises very quickly, and these animals are recognizing pain. The ones that got the P110, the same degree of stimulus, it's like nothing's happened. So not only do they look better, do they have less histopathology, they're actually feeling better as best we can ascertain. Now, I show you this for no other reason than I like this picture. I don't know what it means, but I just think it's very cool. This is Braden. Braden, a clinician scientist. We talked about him in the introduction. He's a real superstar, one of our more junior colleagues. He's got a fancy microscope. He was giving a talk. He knew I was in the audience, so he showed this movie. So this, I think this is gut. It could be liver. This is a blood vessel, and this is a neutrophil, and these little red dots. He stained the mitochondria in a neutrophil, an immune cell, in a tissue that's alive. The mouse is alive. He's imaging it, and you can see the neutrophil just patrols along the blood vessel, up and down, trying to figure out, is everybody healthy? Do you need me? And I'll play it again. And the mitochondria move as well. So they're just remodeling, moving along, fusing and fizzing. So that we saw in a dish, he's seeing in an immune cell, in a tissue. No idea what it means. It's just a cool movie. And I love it. Okay? So to sum up, I think there's loads and loads of evidence that mitochondrial dysfunction is part of IBD, certainly in some individuals. I think that's almost unequivocal. There's lots of evidence in support of that. And the big question is will our discovery research drive new therapies to treat patients with IBD, patients where therapies are really lacking? Because if we can improve the health of the mitochondria, the health of the mitochondrial network, that should improve the health of the cell, and by inference, the health of that individual. And what I've shared with you today is two ways we think this might be feasible in the long run. One is the use of butyrate, which is produced by bacteria. Can we use butyrate or analogs of butyrate to try to maintain or enhance the health of the mitochondria, to enhance that barrier function so that the individual will have less pro-inflammatory stimuli? And then the last little bit there are drugs becoming available for research purposes that says we can manipulate those mitochondria to the benefit of the cell and the animal. And with that, I'll just close by thanking you for your attention, and of course the funders. We have to go after grants, and I've been very lucky with grant support for this. So thank you for your time. Off to the side, sit down. Then going up. Yeah, you want.
So thank you, Dr. Shutt and Dr. McKay, for the fascinating information you shared. We will now open the floor for a question and answer period, and we'll do our best to alternate between questions from our virtual and live audience members. As a reminder, questions can be submitted virtually through Slido by scanning the QR code or by opening slido.com and entering the event code MMS into the participant field. For our live audience, please raise your hand if you have a question, and someone will provide you with a microphone. We will do our best to answer as many questions as we can in the next 10 minutes.
Can you explain the difference between IBD and IBS and the difference between Crohn's and colitis?
Yeah. Online. Yeah, for sure. So IBD, inflammatory bowel disease, IBS, irritable bowel syndrome. IBD, there's obvious inflammation and damage, right? And it's easier to identify. So there's a functional cause. You can see the tissue is damaged. There's lots of signs and symptoms, right? IBS, the colon looks absolutely normal. The patient has horrendous symptoms, often toggling between diarrhea, constipation, bloating, pain, same sort of symptom profile, maybe less bleeding. And the physician with IBS will tend to treat the diarrhea. Unfortunately, most of those patients will go on to have constipation. Then they treat the constipation, and the patient goes back to diarrhea. So a very real functional condition, but no underlying pathology, whereas IBD has different pathologies. Crohn's disease and ulcerative colitis only in the colon, so it never goes into the small intestine. Crohn's disease anywhere from mouth to anus, gum to bum, depending how you remember it, mostly in the colon and terminal ileum. And they look, in terms of histopathology, different. The Crohn's disease can go right through the tissue because you can have inflammation in the thickness of the tissue, get stricturing, fistulas. Ulcerative colitis tends to be more superficial, just that top layer being sort of eroded off. The patients, I'm trying to think of the symptoms, more bloody diarrhea with ulcerative colitis than Crohn's disease. And just so I bring it back to mitochondria, the bug that I showed is associated with Crohn's disease, but some of the original speculations around mitochondria and inflammatory bowel disease was for ulcerative colitis. Going back to 1980, the hypothesis was raised, is ulcerative colitis a metabolic disorder because the mitochondria were damaged?
Right, so there's a question online. What do you know about urolithin A supplements for mitochondria repair?
So I guess I'll start by saying I'm not a clinician. And the quick answer is I don't really know anything about urolithin A specifically as a supplement for mitochondria. Other than to say, generally, we don't really have good therapies for mitochondrial disease that are effective and that we understand exactly how they work. So I haven't heard of it too much before, so I don't think there's anything magical or special about it compared to other treatments in that regard.
Am I live? Can I ask a question? I thought I'd better get this in early because I'm a very dumb person who's got very little knowledge of sitting in the middle of lots of experts here. But I presume mitochondria is the plural of mitochondrium, and there is one effective cell, which is rather like the bacteria that you said infected these whatever they are, complicated cells. So when you're talking about the structure of the mitochondria, are these all within a single cell, a eukaryotic cell, or whatever you call it? Or were you looking at some picture of mitochondria that are extracellular in the body?
So the images you would have seen today are all mitochondria. They're inside a cell. And so, yeah, grammatically, mitochondria is the plural of mitochondrion. And so you're absolutely right there. I guess semantically, it becomes a bit of an argument if these mitochondria can fuse into one big network, or they can divide into all these small parts. Does a cell have one mitochondrion, or does it have hundreds of mitochondria? And I think the answer is it probably depends on that particular cell and how it's feeling at the particular time that you're looking at it. The other aspect, you sort of mentioned mitochondria outside of a cell. There is some evidence that mitochondria can escape cells. And in that context, they can probably promote inflammation. So mitochondria do get out of the cell. We don't really entirely understand how that happens. They're probably not super functional long-term once they're outside of the cell. But there are people looking at ways to transfer mitochondria, get them out of a cell, and then get them back into another cell. And so that's potentially a longer-term avenue for trying to treat mitochondrial dysfunction, is delivering healthy mitochondria to a cell that has dysfunctional mitochondria.
Thank you.
There's another question online. What is the cause of overall general muscle pain associated with mitochondrial disease? Is it driven by an inflammatory response?
I guess I'll try and tackle that. So again, not an expert on the pain or clinical side, but I can certainly imagine there would be an inflammatory component to that. So that wouldn't surprise me at all. Some of it, I think, probably is also going to be linked just to in muscle, specifically the important role for mitochondria in maintaining energy. And if you have mitochondrial dysfunction in the muscle, that can lead to degeneration of the mitochondria, degeneration of the muscle, and that probably also contributes to the pain as well.
So another one from online. What are your thoughts as to why Alberta and Canada are hotspots for, they said IBS, but IBD as well?
Great question. I did say IBD, and there's no answer to that. Lots and lots of hypotheses as to why Canada, Western Europe, Scandinavia have higher incidences of IBD. It's not better surveillance. You say, well, they're not looking for it in China. That's not true. It's a real increase in incidence. Things like vitamin D have been presented. So the further north you are, the less vitamin D you make. Maybe that's linked to it. Others have suggested, well, maybe it's somehow linked to the hygiene hypothesis. So we live in a very sterile environment and we've got rid of other infectious diseases, allowing auto-inflammatory diseases to increase. These are possibilities. Diet has been presented as well, lifestyle. And there certainly isn't a single reason why that's so, although the epidemiology is very clear. It's certainly more IBD in North America, Europe. And when you look at areas with a low incidence of IBD and you start to focus in on the cities, so in South America, there's more IBD in Rio and the developed areas than you'll find in some of the rural areas. So is it this quality of life, westernized diet, high fat, all of these things can really sort of precipitate that. If you think when you go south, over the equator, it's probably not vitamin D where there's not more sunshine. So there are many ways to drive that, but no definitive answer as to why IBD is a global westernized problem, but emerging everywhere.
Right. Are antioxidant supplements good or bad for mitochondrial function?
I want to say good. If you're overproducing the oxidants, they tend to be damaging. As Tim was saying earlier, we need to produce oxygen radicals because they're signaling molecules. They're very important and probably the original signaling molecule if you go back 1.5 billion years. When we overproduce oxygen radicals, these reactive oxygen species, that allows them then to damage DNA, to damage protein, and the cell has a whole raft of endogenous antioxidants to try to control that. So I think the antioxidant supplements would be generically good. Tim might want to contradict that, but good.
So I'll say antioxidants is one of the, I guess, maybe not treatments, but common sort of, I guess, things that mitochondrial disease patients will get. To my knowledge, there's not a lot of evidence that they actually provide any benefit. And there's lots of reasons that could be. Some of the studies, there's just not enough numbers of mitochondrial disease patients to do proper studies. And if you group everyone together as a mitochondrial disease patient, the fact that the underlying dysfunction is going to be different in all these different patients. It could be that antioxidants are good for some patients, but they just don't provide a benefit for other patients. So when you lump that all together, the signal or the benefit sort of get lost in that context. But it's also really important to remember that, as Derek mentions, you need some antioxidant signaling in your cells. So if you have too high a level of antioxidants, you're going to dampen that. And there's actually been some studies in some, I guess, disease organisms where if you block antioxidants, it actually makes things much, much worse. So I just would caution against going too high against the levels of antioxidants. And how do you know how much is too much and how do you bring it down potentially to the right level? So I think we just need to be cautious or careful there from that perspective.
Awesome. So we'll end there. Thank you to our presenters, Dr. Shutt and Dr. McKay, the Snyder Institute for Chronic Diseases, Buxa, AstraZeneca, and the Cummings School of Medicine, and each of you, our audience members, for attending tonight's mini medical school. We hope that you thoroughly enjoyed it and invite you to join us on December 4th for our next mini medical school lecture on how chronic peripheral inflammation causes changes in the brain that may impact conditions like arthritis, colitis, and more. Thank you for joining us and have a great evening.